friends to a new session on genomics and proteomics. In this session, we will discuss DNA sequencing, sequencing strategies, templates, chemistries, labeling and detection, future methods. DNA sequencing. DNA is the information stored that ultimately dictates the structure of every gene product, delineates every part of the organism. The order of the bases along DNA contains the complete set of instructions that make up the genetic inheritance. Sequencing is a key technique in genomics. The term DNA sequencing refers to methods for determining the order of the nucleotide bases adenine, guanine, cytosine and thymine in a molecule of DNA. A wide variety of DNA sequencing technologies have been developed in 1970s, resulting in gains of many orders of magnitude in speed and accuracy. The first DNA sequence was obtained by academic researchers using laboratories methods based on the two-dimensional chromatography in the early 1970s. By the development of dye-based sequencing method with automated analysis, DNA sequencing has become easier and faster. Although two different DNA sequencing methods have been developed during the same period, Sanger's dideoxy chain termination sequencing method has become the method of choice over the Maxam Gilbert method. The DNA sequencing began when Sanger's studies of insulin first demonstrated the importance of sequence in biological macromolecules. The Sanger dideoxy method or chain terminator method, developed by Frederick Sanger and co-workers, awarded the 1980 Nobel Prize in Chemistry, has been dominant in the field. The method relies on replicating many copies of the to-be sequenced DNA segment in 4 separate in vitro reactions, where each reaction is associated with one of the 4 dideoxynucleotides. Instead of a deoxynucleotide, a dideoxynucleotide, that means the analogues of normal deoxynucleotides in which the 3' group or 3' group has been replaced by a hydrogen, is recruited. The replication then suddenly terminates at precisely that base. Hence, each reaction produces many DNA fragments of various length with the same non-base at the end of each fragment. The to-be sequenced DNA sequence can simply be read from the length distribution on the gel using gel electrophoresis. Later, developments modified this protocol to use fluorescently labeled dideoxynucleotides, which permits one reaction containing all 4 chain terminating bases rather than 4 separate reactions. Recently, next generation sequencing technologies based on parallelizing the sequencing process have made it possible to produce thousands or millions of sequences at a cost much lower than those of the traditional terminator based methods.